Have you ever considered the invisible architecture of your daily life? The world you navigate, the technologies you depend on, they didn't just appear. They are the culmination of countless moments of inquiry, of curiosity, of someone somewhere looking at the world and asking, why? This is a story of then versus now, a journey through 7 big leaps in our understanding. It's a tale of small discoveries that rewired science and our lives, from the bubbling hot springs of a national park to the venom of a desert lizard. Our journey begins in a place of primal, earthly power, the geysers of Yellowstone National Park. In the simmering, superheated waters where most life would be instantly sterilized, scientists in the 1960s discovered something extraordinary: a tiny, heat-loving bacterium, a thermophile they named Thermus aquaticus. It was a curiosity, a testament to the tenacity of life in extreme environments. But hidden within this humble microbe was a secret that would unlock the very blueprint of life itself. This bacterium possessed an enzyme, Taq polymerase, with a remarkable talent. It could withstand temperatures that would tear other proteins apart. This heat resistance was the missing key for a revolutionary technique called the polymerase chain reaction, or PCR. Before Taq, copying DNA was slow, tedious, and monumentally inefficient. Scientists had to manually add new enzymes after every heating cycle. It was like trying to build a library one letter at a time, with a new scribe for every page. Let's travel back to the 1930s, a time when our understanding of the atom was undergoing a profound revolution. In the laboratories of Columbia University, a physicist named Isidore Rabi was exploring the mysterious inner world of the atomic nucleus. He wasn't trying to cure disease. He was driven by pure curiosity about the fundamental forces that govern the cosmos. He was listening to the universe on its most elemental frequency, studying how atomic nuclei behave when placed in a magnetic field. Rabi discovered that these nuclei, the very hearts of atoms, could be encouraged to absorb and re-emit energy in a predictable way. He called the phenomenon nuclear magnetic resonance. It was a discovery so fundamental it would earn him the Nobel Prize. For decades, it remained a powerful tool for physicists, chemists, a way to probe the structure of molecules. Imagine a substance that is not quite a liquid, yet not quite a solid. A strange, cloudy, in-between state of matter. This was the puzzle faced by Austrian botanist Friedrich Reinitzer in 1888. He was studying a cholesterol derivative from carrots, and he noticed something bizarre. As he heated the substance, it melted from a solid into a cloudy fluid. And then, at a higher temperature, it melted again, becoming perfectly clear. He had stumbled upon a new phase of matter. Intrigued, but unable to explain it, he wrote to a prominent physicist, Otto Lehmann. Together, they studied these liquid crystals. They were a scientific oddity, a curiosity for physicists fascinated by the phases of matter. For nearly a century, they remained just that, a solution in search of a problem. Chapter 4. Our story takes us now to the sun-drenched salt marshes of Santa Pola, Spain. In 1989, a microbiologist named Francisco Mojica, studying the genome of an archaeon, a microbe that thrives in extremely salty water. As he sequenced its DNA, he noticed something peculiar, strange, repeating patterns, unique spacer sequences. It was a genetic anomaly, a biological curiosity that didn't seem to have any function. For years, he documented these patterns in various microbes, calling them CRISPRs. Mojica became obsessed. He called it his magnificent obsession. He discovered the unique spacer DNA wasn't random at all. It was a perfect match for the DNA of viruses that preyed on these microbes. The repeating patterns were part of a microbial immune system. The bacteria were keeping mugshots, snippets of viral DNA from past infections. Chapter 5. In the arid deserts, southwestern United States, Mexico, lives a sluggish, formidable-looking reptile, the Gila monster. For most of human history, its primary claim to fame was its venomous bite. But within that venom, evolution had crafted a chemical cocktail of surprising complexity. In the 1990s, researchers were studying this venom, not looking for a weight loss drug. They were exploring the frontiers of biology, investigating the potent molecules nature uses for attack, investigating the potent molecules nature uses for defense. They isolated a component called exendin-4. When they analyzed its structure, researchers saw a striking resemblance to a human hormone, GLP-1. GLP-1 is released after we eat. It helps control blood sugar by stimulating insulin production. It also signals to our brains that we are full. Chapter 6. Sometimes, the greatest discoveries come not from success, but from spectacular failure. In 1990, a plant scientist named Richard Jorgensen embarked on a seemingly straightforward project to create a petunia with a deeper, more vibrant purple color. The logic was simple. He identified the gene responsible for producing the purple pigment and inserted extra copies of it into the petunias. He expected to supercharge the color and create a flower of unparalleled richness. But nature, as it often does, had a surprise in store. Instead of becoming more purple, many of the flowers came out completely white, or with bizarre variegated patterns of purple and white. The experiment was a failure, but a fascinating one. It was as if by shouting, the scientist had caused the cell to go completely deaf. By adding more copies of the gene, he hadn't amplified its effect. He had somehow triggered a mechanism that shut down the new genes, and it shut down the plant's own original pigment gene as well. Chapter 7. To find our final discovery, we must look beyond the Earth. We must look to the messengers from the dawn of our solar system. Meteorites. In the 1950s, geochemist Claire Patterson took on one of the most ambitious scientific challenges of his time, to determine the true age of the Earth. He reasoned that meteorites, pristine leftovers from the formation of the planets, would contain a record of the solar system's initial chemical makeup, including its original ratio of lead to uranium. Patterson built the world's first ultra-clean laboratory because he quickly discovered a massive problem. Everything around him was contaminated with lead. The paint, the water pipes, the dust in the air. It was everywhere, interfering with his delicate measurements. Outro. We have journeyed from the microscopic to the cosmic, from a lizard's venom to a rock from outer space. Each of these 7 stories is a testament to the unpredictable, interconnected nature of scientific discovery. A physicist studying atoms has no idea he is laying the groundwork for a medical scanner. A botanist trying to change a flower's color can't imagine he is uncovering a key to new medicines. This is the beauty and the power of the scientific enterprise. It is a journey without a map, guided only by the light of human curiosity. The universe does not compartmentalize itself into biology, chemistry, and physics. These are our labels, our attempts to make the vastness of reality comprehensible. But the real breakthroughs happen at the borders, in the spaces between disciplines, where a strange observation in one field suddenly illuminates a deep mystery in another. The modern world you inhabit is built upon millions of these connections.